Hello everyone, my name is Sophia Stoltz. I'm a PhD candidate in the lab of Brian Husband. Thank you for attending my presentation today entitled Genetic Diversity of the North American Endangered Species Castanea dentata, otherwise known as American chestnut. I'll go ahead first by talking a little bit about why the loss of biodiversity is a worldwide concern. So in Canada, nearly a third of our 3,600 vascular plant species are considered rare. This vulnerability to extinction is influenced by both ecological and genetic factors, such as habitat fragmentation and a loss of genetic diversity. However, we have an overall lack of information on this topic, which is really hindering the development of effective conservation strategies for species at risk. So why restore genetic diversity? First, I'll define genetic diversity as any measure that quantifies the magnitude of genetic variability within a population, including differences in the genetic constitution of individuals and the organization of this variation within and among populations. So one way that we can measure this is using molecular markers such as microsatellites or SNPs. Genetic diversity is the fundamental basis of all evolutionary change. It can be used to infer past demographic change such as bottlenecks and colonization patterns, as well as extrapolate future evolutionary trajectories. The adaptive potential of a population is influenced by the magnitude and form of variation available. A North American species whose populations might be limited by genetic factors is American chestnut. This was once a canopy-dominant tree species that reigned the deciduous forests of eastern North America, from Maine and Ontario west to Michigan and south to Mississippi and Georgia. Its current rarity is attributed to the introduction of chestnut blight at the turn of the 20th century, which completely decimated populations, reducing them to 1 to 10% of their original size. Since then, populations have been plagued by low reproductive success, post-population bottleneck. However, there are reports of infrequent blight-free trees and healing cankers on other trees in Canadian populations, which may indicate greater genetic variation than we previously thought. All right, so for the purpose of this presentation, I will be focusing on the following questions. The first being, how much genetic diversity currently exists in populations of C. dentata? I hypothesize that measures of genetic diversity are low within populations due to the reduced population size that followed the introduction of chestnut blight. And two, does this diversity vary spatially across the range? So I hypothesize that diversity in C. dentata varies in accordance with the central marginal model, which suggests that populations near the center of the range should exhibit higher diversity than those in the periphery due to spatial and genetic isolation exhibited in the northern peripheral populations. So in Canada, our chestnut populations exist in the northern periphery of the range. My samples in Canadian populations originate from a survey from 2014-2015 of about 2,500 trees, where populations are defined as concentrations of trees within a circular boundary of about 25 kilometers radius. So I have 7 populations delineated in red circles in the map on the left, where I sampled 20 to 30 trees per population. And so from each tree, I collected a leaf sample for subsequent DNA extraction. My sampling protocol in the States is very similar, except I sampled along a north-south gradient from the center of the range towards the northern limit at the intersection of the Niagara and Buffalo border. So 6 populations are included along this gradient, where 20 to 30 trees were again sampled per populations, and again leaves were collected for subsequent DNA extractions. Then, with the extracted DNA, I used PCR to amplify primers for 16 microsatellite loci. Then I scored these alleles using geographic Studio. Next, I used a variety of population genetic analyses to measure aspects of genetic diversity within populations, so the number of alleles per locus, the expected and observed heterozygosity, and inbreeding coefficients. I also measured the genetic differentiation among populations using FST and population structure. So here is a table of our standard measures of genetic diversity with the populations represented in each column for the average number of alleles per locus, NA, the observed and expected heterozygosity, HO and HE, respectively, and the inbreeding coefficient, FIS. So at first glance, we can notice that there are high values of expected heterozygosity, but that expected heterozygosity is larger than the observed heterozygosity in all cases. So this slight deficiency in homozygotes is leading to significant inbreeding coefficients in all but 3 populations. Next, I measured population differentiation based on pairwise FST between all population pairs. So pairwise FST ranged between 0.019 and 0.074, which isn't terribly high. However, linearized FST plotted against the geographic distance between populations yielded evidence of isolation by distance, where we have a significant positive relationship here, where populations become more differentiated as the distance between them increases. This brings me to my evaluation of population structure. At the bottom left of this slide, I have an output from a program called Structure, which classifies individuals into larger genetic clusters based on their alleles. So in this figure, each vertical line represents an individual, and they are all sorted by population, with a group of reference samples from the Connecticut Agricultural Research Station of American chestnut trees. The figure on the right has each of my populations represented by a pie chart of the average member of each genetic cluster within each population. So right away you can see this striking difference in population structure between US and Canada, which could indicate some sort of local adaptation or another evolutionary possibility for this organization of population structure. So this brings me to my conclusions and conservation significance of this project. We found evidence of high levels of genetic diversity, although significant inbreeding was present in many populations. We found that genetic diversity does vary spatially, and that genetic structure differs very much between Canada and the US. What does this mean for conservation? So we propose that conservation that preserve and maximize the standing genetic variation through crosses and gene orchards outside the infective range may help mitigate for further loss of diversity and increases in homozygosity due to population fragmentation and isolation. And given the magnitude of genetic differentiation between populations and the distinct population structure across the range, a focus on in situ conservation and using locally collected germplasm is necessary to avoid introducing genotypes that may be maladapted. And with that, I would like to thank Dr. Brian Husband, my supervisor, Paul Crone, Jeff Gross, Amanda Merchant, my lab members, advisory committee members, and friends. Thank you very much.